Typically, cancer research and treatment has focused on shrinking the primary tumor through chemotherapy or other methods. However, most cancer-related deaths are caused when cancer metastasizes, but until last year, there wasn't a specific treatment to target metastases. On May 26, 2017, a study was published that explained the mechanics of cancer metastases and how it could be treated. This discovery has been referred to as the Hasini effect. Cancer metastases is a result of communication between interleukin 6 and interleukin 8. A combination of drugs has been discovered that can inhibit this communication. The purpose of this presentation is to describe the chemistry of the drugs tocilizumab and reparexin. Combining tocilizumab, an anti-rheumatic drug, and reparexin, an inhibitor of CXC receptors, can lead to the prevention of metastases. Our presentation will explain how the chemistry of these drugs interacts with the body. Cancer metastases is the process through which cancer cells break off from a primary tumor, enter the bloodstream or lymphatic system, and travel to other areas of the body. When this occurs, it is typically known as stage 4 cancer, as most people have probably heard of. The blood and lymph systems allow cancer cells to travel far from the original tumor and form new ones, commonly in areas like the liver, lungs, lymph nodes, and bones. Though people can live for years after being diagnosed with this type of cancer, treatment when cancer has spread to this stage usually isn't curative, and metastatic cancer ends up claiming 90% of cancer-related deaths. MMPs, or matrix metalloproteinases, play a central role in the process of cancer metastases. The picture on the right shows the general structure of MMPs. Different MMPs have different structures. Cancer metastases primarily involves MMPs 1, 2, and 9. MMPs are classified as a family of mid-zincan proteases. Proteases are enzymes that break down proteins by the hydrolysis of peptide bonds. This means that these enzymes use water to break down protein polymers into monomers. Specifically, they break down the ECM proteins, or the extracellular matrix proteins. ECM proteins aid in the formation of macromolecules that help bind cells together. They also regulate a number of cellular functions such as adhesion, migration, and proliferation. A portion of the ECM is the basement membrane, which is made up of proteoglycan, fibronectin, fibroblast, and collagen. The proteases also restructure the basement membrane, which provides structural support to the body's tissues and offers input to modulate cellular function. MMPs restructure the basement membrane by the degradation of collagens, which are fibrous proteins. The structure on the left shows the hydrogen bonds in the collagen molecule. Collagen is made up of hydrogen-bound proline, hydroxyproline, and glycine. When hydrolysis occurs, water breaks these bonds by separating the OH- and H+ ions. Many researchers believed metastases happens after the primary tumor reaches a certain size, but Jayatillaka found it was the tumor's density that determined when it would metastasize. Cells reproduce in a confined space blocked by the basement membrane and surrounding stromal matrix. As the cells reproduce, cancer cell density increases. When the human population becomes too dense in an area, we begin to move out to the suburbs or elsewhere to occupy new places. Similarly, when cancer cell density becomes too high, the tumor releases interleukin 6 and interleukin 8, which signals cancer cells to move and spread to other parts of the body. This diagram to the right shows this discovery. When comparing a low density of cancer cells and a high density of cancer cells, it can be seen that as density increases, the amount of MMP expression also increases.
A key component of cancer metastasis is the JAK-STAT complex, in short for Janus kinase, signal transducer, and activator of transcription. The JAK-STAT complex is important in cytokine-mediated immune responses and functions as part of gene transcription and immune regulation. Research in the JAK-STAT field has shown its roles in various cellular processes such as proliferation, apoptosis, and migration, and has also found that frequent dysregulation of the JAK-STAT pathway is common in diverse types of cancer. The complex, like its name implies, is made up of two components, JAK or JAK, which are intracellular tyrosine kinases, and STAT or STAT, which are transcription factors that activate gene expression. The JAK-STAT pathway is activated by the binding of a ligand, such as interleukins, interferons, or growth factors, to specific cell surface receptors, which then convey signals downstream through the JAK-STAT pathway. So how does the JAK-STAT complex relate to the spread of cancer metastasis? Like we mentioned before, the release of interleukin 6 and interleukin 8 is crucial in signaling cancer cells to move and spread to other parts of the body. Interestingly, interleukin 6 and interleukin 8 alone had no effect on cell migration even at high concentrations. Thus, both proteins have to be present in order for the signaling mechanism to work. In fact, interleukin 6 and interleukin 8 need to be at a precise stoichiometric ratio of 5 to 2 respectively to induce cells at low density to move at the high velocity observed at high cell density. The presence of interleukin 6 and interleukin 8 together promotes cell density-dependent migration via the JAK-STAT complex. Cell density-dependent phenotypes are driven by the activation of the transcription factor signal transducer, or STAT3. Increased transcription of STAT3 results in the formation of dendritic protrusions, which then in turn drive cell migration. The JAK-STAT complex is also crucial in the regulation and expression of MMPs. As we know, communication between interleukin 6 and interleukin 8 also promotes the expression of MMPs in addition to density-driven migration. This again is done through the JAK-STAT complex. Interleukin 6 upregulates the expression of MMPs through the JAK-STAT complex, thereby increasing tumor metastasis, as overexpression and activation of MMPs leads to degradation of the basement membrane. Meanwhile, interleukin 8 has been implicated in the upregulation and mediation of MMP release, which correlates with high-grade and invasive tumors. Together, these cytokines promote tumor self-seeding by circulating cancer cells through the increased expression of MMPs via the JAK-STAT complex. When the interleukin 6 cytokine binds to the receptor, the JAK-STAT complex is activated, and thus the cell receives a signal to migrate through the downstream signaling pathway of the complex. The binding of interleukin 6 cytokine also initiates the expression of MMPs, like we just mentioned, which in turn plays a crucial role in the increase of tumor growths. However, when the IL-6 signaling mechanism is inhibited, the JAK-STAT complex will not be able to be transcribed, and thus cells will not receive the signal to migrate. This was the center of Hasini Jayatila's work. When she was a sophomore in Johns Hopkins, she began working in a lab studying cancer cells and was tasked to look at how cancer cells move in a 3D collagen matrix that recapsulated in a dish to replicate the conditions that cancer cells are exposed to in our bodies. Jayatilaka hypothesized that cancer cells are able to communicate with each other and coordinate their movement based on how closely packed they are in the tumor microenvironment. After years of conducting experiments together and merging different ideas and perspectives, her team discovered that the JAK-STAT pathway controls how cancer cells communicate with each other and move based on their cell density. From there, they then decided that they wanted to block the signaling pathway and see if they could slow down the spread of cancer using a cocktail drug consisting of tocilizumab and reparexin.
Tocilizumab is in a class of medical drugs called biologics, which are drugs that are designed to inhibit parts of the immune system that cause inflammation. Clinically, it was designed to treat rheumatoid arthritis by itself, since inflammation is a central feature of rheumatoid arthritis. Its primary method of treatment works, as previously mentioned, by inhibiting the chemical signal interleukin 6, or IL-6. Without tocilizumab, IL-6 connects to the cell and tells the cell to activate. When the cell activates, it contributes to initiating processes that leads to the signs and symptoms of rheumatoid arthritis. The JAK-STAT signaling pathway is a key protein activation in this process and is involved in processes such as immunity, cell division, cell death, and tumor formation. Tocilizumab, therefore, acts as a barrier between IL-6 and IL-6R, which is the receiving protein, which prevents JAK-STAT from activating. This can be seen in the diagram where the orange circles are IL-6, the green block is IL-6R, and the red symbol is tocilizumab. Reparexin is the second conjunctional drug we will be talking about. Its chemical formula is C14H21NO3S. In the diagram to the right are the Lewis structure and 3D model of the drug. In the 3D model, the red atoms represent oxygen, the single yellow is sulfur, the single blue one is nitrogen, and the grays are carbon and the light blue ones are hydrogen. The drug's full name is 4-isobutylphenyl propionyl-methane sulfonamide, and by looking at the Lewis structure, we can determine that the benzene ring in the center and the carbon attachment at the bottom of the structure is the 4-isobutylphenyl, and that the top of the structure consisting of the nitrogen, sulfur, and oxygens is the propionyl-methane sulfonamide. Repirexin is an orally available inhibitor of CXC chemokine receptor type 1, or CXCR1, and type 2, which is CXCR2, with potential antineoplastic activity. Just as a side note, CXC chemokine receptors are actually membrane proteins that specifically bind and respond to cytokines of the CXC chemokine family. Upon administration, repirexin allosterically binds to CXCR1 and prevents its activation by its ligand interleukin 8, or IL-8. This will cause cancer stem cell apoptosis and may inhibit tumor cell progression and metastasis. CXCR1 is typically overexpressed in stem cells, which plays a key role in cancer stem cell survival and the ability of these cells to self-renew. It is also linked to tumor resistance to chemotherapy, which thus, repirexin, when it's in action, helps to inhibit this and will help to battle against cancer. So, by assessing the duality of these drugs, we can determine why these work together to help prevent or limit cancer metastases. Tocilizumab is the drug that works to bind to IL-6R in order to prevent IL-6 from attaching to the same protein, which deactivates the JAK-STAT signaling pathway. Repirexin similarly attaches to CXCR1, which prevents the protein IL-8 from attachment, which deactivates CXCR1 and damages cancer stem cells' abilities to survive. In an experiment performed on mice, simultaneous inhibition of IL-6/8 receptors decreased the expression of WASF3 and ARP2/3 in a mouse xenograft model, which reduced cancer metastases. The elimination of IL-6's and IL-8's ability to connect to receptor proteins disallows cancer cells to receive the signal to migrate, and the cell's capability to renew, duplicate, and spread. This study suggests that simultaneous use of tocilizumab and repirexin could greatly decrease the metastatic capacity of tumors, thereby potentially improving cancer patient outcomes. A similar example of this is how the drug denosumab, which is a human monoclonal antibody that was initially developed to treat osteoporosis, was later found to be effective in the treatment of multiple myeloma and giant cell tumor of the bone. To summarize, the simultaneous use of tocilizumab and repirexin could greatly decrease the metastatic capacity of tumors, which would increase the quality of life for cancer patients and their overall survival rate and outlooks. This discovery has greatly impacted cancer research and has opened up new questions about how we can continue developing this treatment. This includes the development of combination therapies to target tumor growth and metastases, engineering new anti-cancer therapeutics to limit toxicity and to reduce drug resistance, and development of groundbreaking systems that will help for creating better human clinical trials.